Hello everyone. So today we're going to start with the nervous system. Our introductory lecture will look at general classifications, the type of cells that we encounter in the nervous system, and over time we're going to introduce more concepts in neurophysiology, particularly action potential, and then we'll conclude the section by reviewing the autonomic nervous system, the structure of the spinal cord, and finally the structure of the cerebral cortex. The nervous system itself is considered the master controlling and communicating system of the body. It oversees a lot of functions in the body, and it controls them by firing electrical activity in form of an action potential. Now, cells within the nervous system have the capacity to communicate in one of two ways. Either electrical, and that's what action potential is going to be for, or chemical in form of neurotransmitters. These type of communications, they're fairly rapid, they're fairly fast, and they're highly specific, especially when it involves a neurotransmitter, because as you know, neurotransmitters, they have their own receptors that they bind to, and they can cause an almost immediate response within the body. The primary components that we'll encounter within the nervous system would include things like the sensory component, or the sensory aspect, or the sensory division of the nervous system, and that's the part affiliated with collecting information from the environment. That environment, by the way, could be something internal in the body, like when you think of how stretched your muscle is, or it could be from the external environment, things that we see, things that we hear. All of this is a type of sensory information that the body collects. There is usually an integration center. The integration center is going to be something in the brain, kind of a complex interplay between multiple aspects of the brain. The part, for example, if we see something, the part of visual cortex will be involved, memory, and so on. And then the last one would be the motor output. This is kind of the response that the brain will form. That response usually targets an effector organ, and that effector organ could be a muscle. So for example, you see something and then you decide to walk towards it. All right, the fact that your brain decided that you need to walk, it means you're going to activate muscles. All right, you see something and you start to cry. All right, again, your brain decided that this is too emotional, you need to cry. Your tear gland will start emptying, and so on. So here you have the example of seeing a glass of water. So that's the sensory information collected by a sensor. The sensor in this example is going to be the eye, and then from the eye it goes to the integration center, which is the brain. The brain will formulate a response that yes, maybe it's time to pick up that glass of water and drink it if the individual is thirsty. So that's kind of one way of looking at the key players, the key ingredients within the nervous system. In terms of classification, well, the nervous system can be classified in many ways. The two that I'm going to share with you would involve an anatomical classification, which is really based on where the nervous system is, and a functional classification, which will really look at what sort of roles does that part of the nervous system perform. All right. So based on the location, we have the central nervous system, or the CNS for short. Central nervous system includes anything that is housed within a bone cavity. So we have the brain sitting inside the skull, and the spinal cord, which is sitting inside the vertebral canal. All right, the central nervous system is really where the interpretation, the integration of the signal would take place. Now, anything that branches off the central nervous is called the peripheral nervous system. All right, so the peripheral nervous system is really any division, branch off the central nervous system. The branches, the nerves that originate from the brain, are called the cranial nerves, and the ones that originate from the spinal cord are called the spinal nerves. All right, so the brain and the spinal cord, they make the central component, or the central nervous system, and their branches, whether it's the cranial nerves or the spinal nerves, that will make the peripheral nervous system. So that's location, all right? And the in the bony cavity, usually towards the midline of the body. That's central. Anything that branches off and moves towards the periphery, that is considered peripheral nervous system. All right. The next classification is the functional, the physiological classification. All right, and that looks at function. What sort of role does that part of the nervous system play? You can see the two primary categories. We have the sensory nervous system, which is, as I said earlier, is the collecting part, the part that will pull information from the environment to the brain. We also call this part the afferent. Now, if you think of the brain being the integration center, there is an input to that brain. That's really the sensory or the afferent. And there is an output from the brain, which will be the motor or the efferent. Now, students often confuse the term afferent and efferent and try to remember which is which. The easiest way to do it is that the E in efferent is for E in exit. Exit is always away, all right? Or always out, if you wish. So anything that leaves the brain, moves away from the brain, is an efferent type of information. So the sensory component could be something that we are fully aware of. So we call that somatic sensory. So anything that relates to, for example, you touch a surface and you feel that it's too warm or too cold, that is something you are fully conscious of. So that will be the somatic part. Or it could be visceral. Visceral, it means it's basically your organs, your internal organs. So you may not be fully aware of it. So how does that work? Well, your brain will be notified. Your brain will make an interpretation and then formulate a response, but you don't know about that response. All right, and we call that the visceral. The same, or the second division, is the motor. Motor is the output. It's whatever the brain decides. And it's the efferent. Remember, the E in efferent for E and exit. This will transmit impulses from the CNS towards the effector organs. The effector organs, if you recall, they're going to be muscles, either the skeletal muscle, the smooth muscles, or the cardiac muscles, and all the glands in the body. Again, the motor response could be something you are aware of. So if you decide to get up right now from your chair and start walking, well, that's because you've activated the somatic nervous system to trigger your skeletal muscles. If you had dinner before watching this video or a meal, then your brain will fire up the smooth muscles in your stomach to start digesting the food. All right, but this is something you're not fully aware of, so it's part of the visceral nervous system, which is really depicted here as the autonomic nervous system. Autonomic nervous system, this is the motor part of the unconscious system or the visceral system, and it has two arms, sympathetic and parasympathetic. And this is something we'll discuss in a future lecture. So the somatic motor, it conducts impulses primarily to skeletal muscles. It's voluntary, it's conscious, you are aware of it. The visceral, on the other hand, it regulates three things. Smooth muscles, so think of your hollow organs, your gut, your stomach. Cardiac muscle, so the heart, and glands. Those are things that you are not really aware of. It is involuntary, and as I said, the term autonomic nervous system is an umbrella term. It covers both, or it has two arms, sympathetic nervous system and parasympathetic nervous system. Now, the one definition I will give you at this early level is that sympathetic and parasympathetic, they oppose each other. Whatever sympathetic does, parasympathetic will do the opposite, and vice versa. Now, depending on your academic background, you've probably taken a biology class or an A&P class, and you've probably learned something that sounded like this. Sympathetic always elevates parameters, elevate things, speed up things. Parasympathetic always slow things down, inhibit, suppress things. That's not really an accurate definition or distinction between the two. The more accurate one, like I said, is that they oppose each other. All right? Whatever one system does, the other system will do the exact opposite, and so on. All right, so let's look at these classifications in a picture now. So as you can see, everything in red, the red arrows, they represent the motor output of the nervous system, and anything that you see a blue arrow next to it will represent the sensory part of the nervous system. All right, so the motor system, we said we have the somatic, the conscious, which as you can see is going primarily to skeletal muscles, and we have the autonomic. All right, and autonomic, if you recall, we said the target for it is either a smooth muscle, a cardiac muscle, or a gland. Here you're seeing a branch going into the heart, a branch going into the smooth muscles of the bladder. So smooth muscle. And then the sensory input. There is sensory input from the skin, something you are aware of, whether it's touch, it's pain, temperature, or input from the stomach being a visceral organ. So it could be, for example, change in how much the stomach is stretched. This is something you are not aware of, but your brain is aware of. And that's how the brain initiates digestion within the stomach. All right. So now that we have a general understanding, a general idea of what the divisions, the classes, the classifications are, we can move on to the histology. Now, the way we're going to approach histology is we'll resume the discussion we started about a week ago when we were dividing the tissues, looking into their criteria, and so on. So, from a histology or microscopic standpoint, the nervous tissue is highly cellular. It doesn't really have a lot of extracellular space. It's tightly packed with cells. And there are two types of cells. We have the actual neurons, and if you remember, the neurons, they are the building blocks, the units, the nerve cells. They are excitable. They have the capacity to transmit electricity. We also have the neuroglia or neuroglial cells. Neuroglial cells, which is kind of like the same name. Those are the support cells. All right, they provide support to the actual neurons. All right, now the neuron, we'll conclude our discussion later on by looking at the neuron, but for now, let's focus on the neuroglia. The way I'm going to present those neuroglial cells is that I'm going to divide them into two groups. We're going to look at cells, neuroglial cells, support cells, that we only see in the CNS. So we'll have 1, 2, 3, and 4, and then 2 cells, neuroglial cells, that we'll see in the peripheral nervous system. So I'll go over their names, and then after that we can start looking into what each cell is, what does it look like, what sort of function does it perform, and then I'll show you a picture of it as well. So the first one, the most abundant one, is called the astrocyte. Astro means star, cyte is the mature cell. The second one is microglial cell. Third one is ependymal cell. Fourth one is oligodendrocyte. Fourth one is satellite cell. And the last one is the Schwann cell. All right, as I said, those are only in the peripheral nervous system, everything else in the central nervous system. So let's start with astrocyte. Now, they are the most abundant. They are highly branched, hence the name astro, so they have that star shape. And they have the capacity to cling, to attach to two things, either the neurons or the capillaries. All right. Now, if you recall, in a previous discussion, we did compare one particular cell to that of a star shape. Do you guys remember what cell was that? If you're thinking of the osteocyte, you're right. Osteocytes have that star shape, and it is highly branched as well. What sort of roles does astrocytes perform? Well, it provides support, it plays a role in the exchange. Remember, because they are clinging into capillaries, so now you have the wall of the capillary, so that's kind of one layer, and then you have the neuron wall. So now you have like two walls pressed against each other. When that happens, they will make a filter, all right, or a barrier if you wish. That barrier is called the blood-brain barrier. All right, so that barrier will control what flows in and out of the brain. The general rule is that anything that is lipid soluble and small will pass through that barrier. Anything that is large, so has a large molecular weight, that is water soluble, will not pass through that membrane. All right, so why is it there? Well, I'll remind you that the brain or the neurons are vulnerable cells, meaning that they need the extra layer of protection, so we need that barrier. Other roles that astrocytes perform is that they guide migration of junior or young neurons. They control the chemical environment because they control what goes in and out from the blood. And they influence neuronal functioning to a certain extent. That's not their primary job. All right, so here you can see the astrocyte. Again, it's a star-shaped cell, so it has all these branches, and it's clipping on two things. It's clipping on neurons, and it's clipping on or clinging on a blood vessel, a capillary. So now this is the wall, this is the wall, now you have a filter, and that's the blood-brain barrier. All right. The second type of cell that we have is the microglial cell. They are small, hence the name micro. They're ovoid, and they have thorny processes. All right. Now, they have the capacity to migrate towards injuries, so any injured neuron, and they can phagocytize any microorganism or cellular debris that end up in the nervous tissue. All right, and because they have the thorny processes, it means they could basically grasp something and eat it up. Now, does that function remind you of anything? If they have thorny processes, they chase things, they kill things and eat things. What do they sound like? If you're thinking a macrophage, you're right. What I would encourage you to do is to associate the M in microglial cell with M in macrophage. All right. So if you recall, about a week ago, I've mentioned to you that macrophages, will encounter them a lot this term, but we'll see them under different names. So here, microglial cell, it's the actual macrophage of the brain. When we discussed the skin, we've encountered Langerhans cell, and that was the macrophage of the skin. All right. But functionally speaking, it's the same function. It eats, it cleans, injuries and debris. All right, so this is what it looks like. Look at all these small thorny processes, which could actually trap small bacteria somewhere, and then the cell will eat it up. All right. The next cell is the ependymal cell. And those range in shape. They're kind of like epithelium. They go from flat squamous to columnar and anything in between, so the cuboidal. They may have cilia, and we know cilia exist on the apical surfaces. Cilia has the capacity, in this context anyways, to circulate the cerebrospinal fluid. Cerebrospinal fluid. All right, and they line central cavities within the brain and the spinal cord. Why? Because that's where the CSF is. Now, why do we need them? Well, let's look at a picture first and then try to understand their anatomy and their role. All right, so this is a picture from a fluid-filled cavity in the brain. It could be a ventricle, in the cerebral cortex. You can see the ependymal cells. Here they are kind of like cuboidal, 2-columnar, and you can see the cilia on the top. All right. Now we have this fluid here, the cerebrospinal fluid. Problem is, when you have fluid that has a temperature of 37 degrees, that has some sugar in it, if it's sitting for a long period of time, it becomes an attractive medium for bacteria, all right, for bacterial growth. So one way to eliminate that is to keep that fluid rotating, circulating, all right? So what these cilia do, they basically, remember, cilia, they have wavy movement. They go back and forth. All right, that sweepy back and forth movement will keep that fluid circulating. All right, and when the fluid circulates, it makes it harder for the bacteria to colonize and to replicate in there. All right, the next cell is the oligodendrocyte. It's a highly branched / efficient cell. Oligodendrocytes, they are in charge of wrapping the neurons of the central nervous system. Is that important to remember? Yes, simply because the neurons of the central nervous system are myelinated by a different cell compared to neurons of the peripheral nervous system. What sort of wrap are we talking about? It's called the myelin sheath. Myelin sheath is a lipid protein material that will act as an insulator of nerve cells, and more importantly, it will speed up nerve conduction. All right, we've put this myelin around CNS neurons thanks to oligodendrocytes, and put it around peripheral nervous system neuron thanks to a completely different cell, which I will outline in the next couple of slides. So this is what an oligodendrocyte look like, highly branched, and here you have 6 neurons or nerve fibers, all being wrapped at the same time. That's how efficient oligodendrocytes. Oligodendrocytes, they're actually efficient to the point where they can wrap or myelinate 60 neurons at the same time. That's really efficient. Now, the cells in the peripheral nervous system, we had 2 examples. We have the Schwann cell and the satellite cell. Schwann cell is the cell that will make myelin sheath, but for peripheral nervous system neurons only. Because we've learned recently that the central nervous system neurons, they are myelinated by the oligodendrocyte, so by a completely different cell. Satellite cell is a support cell. So let's keep it as simple as that for now. It's not a high-yield cell, not like the other ones that we've discussed. All right. So this is basically a review of the criteria of the key cells, at least the neuroglial cells, that we see in the nervous system. The next part of our discussion will be dedicated to the primary building block. What is that building block? Well, it would be the neurons, the actual neurons. Now, neurons, they are the structural building blocks, units of the nervous system. They are large. They are highly specialized to transmit impulses. They have extreme longevity. They could live for 100 years or longer. They are amitotic, so that's the opposite of mitotic. What does that mean? It means they have lost the capacity to divide. If they don't divide, it means if they die, we don't have the capacity to make new ones, so they're gone forever. There are a few exceptions, but that's a discussion for a different class, so we'll leave that out. They have high metabolic activity, and that's actually where their weakness comes from. It means they require a lot of oxygen and glucose to work. So because they are really dependent on high levels of oxygen and glucose, the moment you deprive them of oxygen and glucose, they start to suffer greatly, and they could die right away. They have a cell body, just like any other cell that we've encountered, and they have one or more processes attached to that cell body. Some of these processes, as we'll see, will serve as input. Some of them will serve as output. All right. So the cell body itself, also known as soma or perikaryon, it's the actual biosynthetic center. It makes proteins, it makes membranes, other chemicals. There is the rough endoplasmic reticulum, and it has a nucleus, a nucleolus. Some may contain pigment cells. And in most neurons or cell bodies, the plasma membrane part of the receptive region. Now, when we see nerve cell bodies accumulating in the nervous system, central or peripheral nervous system, we use specific terminology. So every time we see nerve cell bodies in the central nervous system, we use the term nucleus for one, or nuclei. Keep in mind that this term, nucleus or nuclei, in the nervous system context has nothing to do with the nucleus of a cell. Here we're just saying that if we have a group of nerve cell bodies in the CNS, we call them nuclei. That same collection, that same cluster of nerve cell bodies, if it happens to be in the peripheral nervous system, we don't call it nucleus. We call it ganglion for one, or ganglia for multiple. All right, so those two terms, nuclei and ganglia, they are basically specific terms for nerve cell body collection, either in the central nervous system or the peripheral nervous system. All right. So let's look at a picture of that. Let's see how it looks like. We've seen a similar picture in the past. We have the actual nerve cell body to the center. We have the nucleus. If I zoom in further, you can see the endoplasmic reticulum, the mitochondria. So all the organelles that we've encountered before can be seen in here. We have multiple input sites, and these are called the dendrites, or the receptive regions. And we have one large output site, which is the axon. The point at which the axon emerges from the nerve cell body, so that kind of funnel-shaped region here, is called the axon helix. All right, it has a name. In most cases, a nerve cell has multiple dendrites, so multiple input centers, but one large axon, one large output center. All right, and that output center will go into the target organ, or the cell, and it has all the axon terminals. Anything could be in this region. A skeletal muscle could be in here, and that will make the neuromuscular junction, if you remember this from our last lecture. So the dendrites, there are hundreds of them in a neuron. They're fairly short, diffusely branched processes. They are the receptive regions, the inputs. They convey incoming messages towards the cell body in form of graded potentials. Graded potential is something we'll talk about on our next lecture, but in summary, a graded potential is a local current. Remember, electricity in the nervous system, it has to start somewhere. It starts as a local current and then it travels somewhere else. So when it begins, when it starts, it's actually a graded potential. All right. Now, the collecting point or spot on these dendrites is called a dendritic spine. which is kind of like a spiky end within a neuron. I'll show you a picture of an electron microscope picture of a neuron just to show you where the spiky end is. All right, so here you can see the actual cell and all the branches of the cell. All right, so this is one dendrite. If I'm looking closer on that dendrite, I see a spine, all right, a thorn. That thorn is basically the dendritic spine, which is really where the collection part of information would begin. All right, the next component is the axon. The axon, there's one per cell, and it exits through the axon helix. It could be very short or absent in some cells, and in others it could be as long as 1 meter. Now, they terminate at what's called the axon terminals, and that's where we reach a particular cell or a target and basically convey the information to that target. The axon has its own membrane called the axolemma. This is kind of like saying the sarcolemma in the muscle. There is a secretory region, and the neurotransmitter are released from their storage vesicle. This is kind of very similar to when we started our discussion of muscle contraction. We said acetylcholine, the neurotransmitter, is stored in little vesicles, and those are released when there is electricity. There is no ER, no Golgi apparatus in the axon. So how does the axon survive? Well, it relies on the cell body to receive new proteins and membranes. So it doesn't really synthesize what it needs. It relies on incoming cargo from the cell body. There is a fairly efficient transport mechanism, and if axons are cut or damaged, they will decay right away. Now, when you think of the relationship between the axon and the nerve cell body, so if I say nerve cell body and the axon, we're either going to carry information towards the nerve cell body or away from the nerve cell body. If that's the case, every time we carry something back to the nerve cell body, we call that retrograde movement. Anytime we move things away from the nerve cell body, we call that anterograde movement. All right, so anterograde is away from the nerve cell body. What sort of things move that way? Mitochondria, cytoskeleton, enzymes, and so on. What moves in a retrograde? Well, it could be organelles that are ready to be eliminated, degraded, worn out organelles, for example, any signal molecules, and any microorganism, foreign invader that is trying to exploit the nerve cell, a virus, a bacterial toxin, and so on. This is, by the way, retrograde travel is how the rabies virus move. All right, remember, rabies virus, you can get it from being bitten by a wild animal that's infected with the rabies virus, and then the virus will slowly move through the nerve fiber all the way back to the nerve cell body of the central nervous system. That's why people who are infected with rabies, most of the symptoms they develop are nervous system symptoms. All right. The last of the discussions we have on our agenda will be dedicated to the myelin sheath. Myelin sheath is really the myelin, the protein lipid substance that wraps the neurons. When neurons are surrounded by that myelin, we call them myelinated fibers. When they're not, we call them non-myelinated or unmyelinated. Keep in mind that even with myelinated neurons, they're not all myelinated the same way, meaning that there are heavily myelinated neurons and less myelinated neurons. So really it's kind of like a spectrum. Heavy myelinated neuron, less myelinated neurons, and unmyelinated neurons. So it will encounter a wide spectrum of variations. What does that myelin do? Well, it protects, and so it protects the actual axon and provides electrical insulation. And that will increase the speed of nerve transmission. So that means heavy myelinated neurons, they will have the fastest conduction speed. All right. When you think of the nerve responsible for vision, so your optic nerve, all right, optic nerve is the most heavily myelinated nerve in our CNS. That's why sight or vision travels really fast. We see things before even hearing them. So that means the nerve carrying visual information is more myelinated than the nerve carrying auditory sound information. So we see things faster than hearing things. So how does it form? Well, if you recall, there are different cells that myelinate neurons. In the peripheral nervous system, we have the Schwann cell. Schwann cell is not really a very efficient cell. It clips around a neuron and it starts to wrap itself in layers, concentric layers. So every time it rotates, it adds a layer of that myelin. So it's kind of concentric layers, looks something like this. All right, so let's look at a picture. First thing that has to happen is for a Schwann cell to find a nerve, an axon, clip into it. And then that cell will start to rotate. So now we have multiple layers. And then once it's done, it's basically done. It adds all the myelin sheath that is needed. And now another cell will clip on an adjacent part of that neuron and start adding more and more layers. So here we can see an actual cross-section in a nerve. So we're zooming in 24,000 times to see this nerve. If you zoom in with your notes, you can see there are layers, concentric layers. Each layer is actually myelin sheath. The more layers we see, the more myelination there is, and that means the nerve will have a faster conduction speed compared to others. All right, so in the exam, hypothetically, if I show you two pictures, one of them has, let's say, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, roughly around 14 myelin layers. And I show you another nerve that has, let's say, 1, 2, 3 myelin layers, and then ask you which one conducts faster. Well, the more myelination, the more layers you see, the faster the conduction speed is going to be. So that's the story in the peripheral nervous system. How about the central nervous system? Well, central nervous system, the myelin is formed, if you remember, by the oligodendrocytes. All right, oligodendrocyte is way more efficient than the Schwann cell. Each cell, each oligodendrocyte, can wrap up to 60 axons at once. So that tells you how efficient and how good it is at multitasking. In the central nervous system, we see variations in the tissue. We see the white matter, which is really the area where we see a lot of myelinated fibers. That's why it appears light, it appears white, because of all the myelin. Remember, myelin is a whitish protein lipid substance. And then the gray matter of the brain, it's actually the opposite. It looks a bit darker. Why does it appear dark? Because it contains the non-myelinated fibers. So that means it's not as going to conduct things as fast. All right. Now, in future discussions, especially when we look at the spinal cord and the brain, both being central nervous system, I'm going to remind you of the white matter and the gray matter. Now, structurally, they're the same. White matter, the light areas, is where all the myelinated neurons are. Gray matter, the dark area, is where all the non-myelinated neurons are. Are there any differences between their locations? Yes, and that's where it gets a little bit interesting. So we have the spinal cord, we have the brain. Both are central nervous system. Now, the white matter and gray, white matter and gray. The white matter in the spinal cord lies to the outside, whereas the gray matter of the spinal cord lies to the inside. So this is superficial and this is deep. Where do you think it's oriented in the brain? It's actually the opposite, and that's where the fascinating part about how our brains are made is. The white matter is deep and the gray matter is superficial. All right, and then once we get to those sections, I'll provide more details about functions. So the white matter in the spinal cord, for example, is basically where all the tracts are located. So motor tract, sensory tracts. Gray matter, on the other hand, is where the sensory cells, nerve cells are, as opposed to motor nerve cells. And the brain, the white matter, the deep matter, is really deep nuclei. These nuclei could be in charge of regulating how you move, regulating a lot of things about our biological functions. But we'll cover those in their respective sections. All right, so I'll stop here. Next time I'll see you, we'll start with the next set of the introductory lecture, which will primarily focus on action potential. So how are action potentials generated? How are they transmitted? What are the phases of action potential? And what is refractory period? That would be the main theme for the rest of this lecture.